Welcome, everybody. Good morning, or good afternoon, or good evening, depending on where you are. I'm Christine from Nature Reviews Materials, and together with Chiara from Nature Nanotechnology, we want to welcome you all to this webinar and Q&A session on lipid nanoparticles for mRNA delivery. We're super excited that so many of you could join us today from really all over the world. And in this webinar today, we want to talk about the technology behind the new COVID mRNA vaccines, such as the ones developed by Moderna and Pfizer-BioNTech. So the technology or the idea of using nanoparticles, and in particular lipid nanoparticles, for the delivery of drugs and nucleic acids such as mRNA has been developed and investigated and optimized for many years by the nanomedicine and material science communities. So we actually know a great deal about this technology, and we are very lucky today to have two experts in this field with us, Kathryn Whitehead from Carnegie Mellon University and Yizhou Dong from Ohio State University. Welcome you both, and thanks so much for joining us today. So Katie Whitehead is an associate professor and Dean's Career Fellow in the Departments of Chemical Engineering and Biomedical Engineering at Carnegie Mellon. And her lab develops RNA and protein drug delivery systems, really with the long-term goal of predicting and investigating the behavior of these materials in humans. And Yizhou is associate professor in the Division of Pharmaceutics and Pharmacology in the College of Pharmacy at Ohio State. And his lab, his research focuses on the design and development of biotechnology platforms for the treatment of diseases, including infectious diseases and cancer. And he has worked with mRNA therapeutics for quite a while. So Katie and Yizhou are going to kick off our webinar today with two short talks in which they will explain the technology, tell you how these mRNA vaccines work, what they consist of, and they're also going to provide a bit of a historical context, basically of what's been done previously with this technology already. And after the two short talks, we will then try to answer as many questions as we can that you may have. Many of you have already send some questions during registration, which is great. Thanks so much. And we will definitely get to those questions, but please also use the questions box that you can see on your right-hand side, hopefully, in the GoToWebinar application to just type in questions throughout the talks, throughout the webinar, and we'll try to answer as many as we can. And then without further ado, I think I'm going to hand over to Katie, who's going to kick off the webinar today. Thank you, Katie.
Thanks, Christine. It's so nice to be with you all today. And during my presentation, I'd like to teach you a bit about these mRNA-loaded lipid nanoparticles that are doing such a wonderful job in vaccinating populations against SARS-CoV-2. So I'm going to explain a bit about the drug itself, so the medicine itself, which is the mRNA, as well as explain how we put it inside a lipid nanoparticle and what exactly that lipid nanoparticle is. So let's see if I can advance. Okay, so messenger RNA, so mRNA, this is a genetic molecule for those of you who are not familiar, and it's part of the process in our bodies that makes proteins. So what you're seeing here is called the central dogma of molecular biology, and it tells us that we start with the blueprint information, our DNA, which is permanently stored in each one of our cells, and this DNA is converted inside of our cells into messenger RNA. And you can think of messenger RNA as a little bit of a middleman, relaying the information that the DNA is giving us and using it to create proteins. So proteins are the doers in our body. They're responsible for nearly all functioning within our bodies. And so really any kind of disease or condition, it almost always has some critical protein involved. Either there's too much of a protein, too little of a protein. Sometimes a protein has been mutated and so it doesn't have the correct shape or form or function. So we're excited about messenger RNA as a type of therapeutic because it's upstream of this protein production. And because it's a middleman, because it's temporary, we can actually try to fool around with its expression so that we can make changes in protein without changing our DNA, without permanently altering anything in our DNA. So one of the ways that we can do this, if we want, for example, more of this blue protein here, we can simply add more of this same messenger RNA. So the messenger RNA has a particular code. We can add more of it into our bodies, and so we can augment the expression of that particular protein. Similarly, if we want to make some other protein, perhaps even a protein that our body doesn't even normally know how to make, we can deliver a different sequence of messenger RNA into our body and ask our bodies to make that protein as well. And so in this way, we can induce all sorts of protein expression that can help either treat or, in the case of these vaccines, to prevent disease. So just a little bit on what mRNA drugs can do. So vaccines are clearly at the forefront of applications that are being used in the clinic. I've been working in the area of RNA delivery for over 15 years now, and as with many of my colleagues, we've long been believers of the enormous potential of these genetic medicines. And let me just explain to you a few of the different types of conditions that can be treated with messenger RNA. So one of the most obvious types of applications is protein replacement therapy. So for example, those with hemophilia, this is a blood clotting disorder. And the reason people bleed is because they are missing a particular protein in our blood that helps the blood clot. And so a fairly straightforward application of messenger RNA would be to get that messenger RNA sequence for that missing protein, to deliver it into the body, and then our body would be able to make that protein and the blood is able to clot. Okay, so there are numerous different conditions where we need to make more of a protein or to introduce a missing protein into our system. Vaccines, as I mentioned, have been the most clinically advanced application. And in this case, we're not replacing any kind of missing protein, but instead we're introducing a type of protein into the body that our bodies have never seen before, a foreign protein called an antigen. So in this case, for SARS-CoV-2, we're introducing a small protein found on the viral surface. And when we show that to our immune system, after the cells create that small antigen, the immune system reacts in a way that it will help us respond to any kind of infection that might take place. And so to prevent that infection or to prevent a very severe response to that virus. I won't go into them in a lot of detail, but messenger RNA similarly can be used for cancer immunotherapy applications. So we can deliver messenger RNA that encodes for proteins that will help teach our immune system how to attack the cancer cells in our bodies. We can also use messenger RNA to create what are called transcription factors. These are a type of protein that help the stem cells in our body decide which other types of cells they're going to become. So we can help our stem cells create new types of tissues using this technology as well. And then finally, for those of you who are aware of gene editing or interested in it, we can actually turn down protein expression. All these other ones involve creating more protein expression, but we can turn it down by delivering messenger RNA encoding for a nuclease called Cas9 or any of the other nucleases, which can then go into our nuclei and carry out gene editing to silence particular proteins in our system. And if any of you are interested in learning more about details of these applications or to learn more about the delivery vehicles that are being used, you can reference this review paper that a member of my group wrote a few years ago. So the problem with messenger RNA as a therapeutic, we can't just take a pill, we can't just inject it into our systems and hope for the best. This is because it's a very large molecule and it's negatively charged. our cell membranes are also negatively charged. So when those two negative charges come up against each other, the mRNA is repelled and it's not able to enter the cell. If we just inject it into our bloodstream, it will be cleared very rapidly through our kidneys. And so what we really need is some kind of a delivery vehicle that's able to package up this messenger RNA and then take it to where it needs to go inside the body, into the cells where the protein expression is needed. So I wanted to just put together a slide here that would highlight all the different things that we need this delivery vehicle to do. It's really, in my opinion, pretty miraculous that we can come up with something that works. So in the case of vaccination, we are going to receive an injection of these lipid nanoparticles into our muscle tissue. Okay, so those lipid nanoparticles then need to diffuse to the site of the cells that are going to be vaccinated. Sometimes these cells are going to be the muscle cells, sometimes they might be the immune cells, because there are also immune cells that reside in our muscle tissue. One way or another, these lipid nanoparticles will make their way to the vicinity of these cells, and they're taken into the cell through a process called endocytosis. So basically, the cell membrane reaches up and around the nanoparticle and pulls it inside. Normally, the cell does this to try to sample its environment and to find nutrients and other molecules that may be out there that it wants. When it brings in a lipid nanoparticle, it doesn't know what to make of it. It's in this walled off compartment called the endosome. And the cell keeps it there because if anything damaging is brought in, it wants to be able to get rid of it. So the problem that these delivery vehicles face is that they don't readily exit this endosome. So the chemistry of these particles has been designed very carefully over the years to enable this endosomal escape process, as we call it, which will then allow the lipid nanoparticle to deliver the RNA into what's called the cytoplasm of the cell, which is the watery area outside the nucleus. And that is where the proteins can then be made. So really, we're asking our nanoparticles to do quite a lot, overcome a number of different barriers. And it's really decades of research that has enabled us to get to where we are today and to the very rapid development of these vaccines. So cationic lipids have been used for the earliest forms of nucleic acid delivery. So DNA is where researchers really started in this field. And DNA, although it's structurally somewhat different than RNA, it's still made up of very negative nucleobases or nucleosides. And it also, it's a very large molecule. And so some of the properties that we need to encapsulate DNA are very similar. So what researchers decades ago found was that they could use cationic lipids, so these lipids that have a positive charge, and then here is something like a 16 carbon long tail, two of them. And if you're able to use these these lipids to create liposomal structures that encapsulate the DNA. So here's what a liposome, a traditional liposome looks like. This is a cross section right here, and here's what it kind of looks like in 3D. So it's similar to our cells. It has a lipid bilayer all around the exterior of the liposome. And these blue balls here represent that cationic head group on the lipid, which is water-loving. So this water-loving group faces outward into an aqueous solution. And then we also have these water-loving materials facing inward. Meanwhile, the tails all come together because they are hydrophobic and they want to hang out with each other. So inside, the DNA, which is water-loving, can be encapsulated. Over the years, the technology and the exact chemistry used for nucleic acid delivery has evolved. And DNA, as I mentioned, it is a little bit different than RNA. And so as we've moved into RNA delivery, we've also had some developments in the type of particles that are used for efficient RNA delivery. So we're now at a point where these SARS-CoV-2 vaccines, they're making use of more recent forms of lipid nanoparticles. These are a bit more of what I'd call a solid lipid nanoparticle, and they're made up of several ingredients. So one is called an ionizable lipid, So it's like that cationic lipid I showed you before. However, it only takes on positive charge inside the cell. And because it's neutral in the bloodstream, it actually causes less toxicity than some of the earlier versions of these lipids. There's also a fair amount of cholesterol in these particles, just the same way that there's a lot of cholesterol in the cell membranes inside our body. So it serves a similar purpose where it helps to fluidize our particle to provide some support and shape to the particle and stability. And then there are what we call helper lipids. These are phospholipids, also can be naturally occurring lipids that we incorporate into these particles that can help with the packing. So you can see all these different molecules are packed inside this particle. This is just my kind of interpretation for what might be going on here inside the particle. These are very small particles, and so to my knowledge, you know, we only kind of have clues and bits and pieces as to what these look like on the inside. But the specific helper lipid is chosen to help this messenger RNA shown in black to really pack in there effectively. There's a molecule called polyethylene glycol, or PEG. This is a polymer that is currently found in some FDA-approved medicines, and this hangs out on the exterior of the particle. It also helps with the stability of the particles, and for other applications, it can help shield from unwanted immune clearance, although that's not as much of a concern in the case of vaccines. So this is what these particles look like. My lab works with these particles. Yizhou's lab works with these particles, many different types of very similar looking particles. They're all very similar. One thing that tends to change is the ionizable lipid. So this is where a lot of the intellectual property exists within the lipid nanoparticle space, and it is the major difference between the Moderna and the Pfizer BioNTech vaccines. Okay, so here are these ingredients again. Both of the vaccines use the same set of 3 other lipid nanoparticle ingredients. They all use cholesterol, they all use a helper lipid called DSPC, and they all use a very specific version of this PEG, okay? Their messenger RNA sequences are a little bit different, but essentially are going to do very similar things once they get into the body. As for the ionizable lipids, this is what BioNTech Pfizer's ionizable lipid looks like. So you can see here, it's a neutral molecule. This is what you know, the muscle tissue sees until these molecules make it inside of the cell. And this amine group right here is able to take on a positive charge that helps our nanoparticle escape that endosome, okay? So it's very interesting that if we look at the Moderna lipid, this is a very, very similar looking lipid, right? So we have an alcohol group. The distance between the alcohol and the amine is a little bit different. It's 2 carbons over here versus 4 carbons over here. And then we have similar tail structures. All of the tails incorporate this group right here. This is called an ester, and this ester group is degradable under the conditions inside of our body. So this helps to make sure that after our lipid nanoparticles have done their job, they're able to break down and to degrade inside of our body so that they can be cleared and they don't accumulate to cause toxicity. So all of the tails have this. And then one of the other differences here is that we have essentially 4 short tails at the end of the BioNTech Pfizer ionizable lipid, and we have 3 tails over on this end. So structurally very similar lipid nanoparticles. So I mentioned before that RNA is negatively charged, and these ionizable lipids, they become charged only under reduced pH conditions. So we put these materials into slightly acidic solutions in order to form the nanoparticles. So these electrostatic interactions help draw our materials together. And you can make the nanoparticles either by hand using pipettes, or you can make them in a microfluidic device. There are a number of different ways, but essentially you're going to be mixing an RNA stream, which is an aqueous stream in water, and you have a lipid mixture, which is dissolved in ethanol. And when you rapidly mix these two streams together, you get lipid nanoparticles, which precipitate out of solution. They tend to be about 80 nanometers in diameter, but that can vary depending on the exact ingredients that you put into that nanoparticle. So just a couple more slides from me explaining how these messenger RNA vaccines are helping protect specifically against SARS-CoV-2. So this is a depiction of SARS-CoV-2, the virus. And on the surface of the virus are what are called the spike proteins. You can see these kind of look like a crown or a corona, so that's why they're called coronaviruses. And this is how the virus, the spike protein, is how the virus enters our cells. So this spike protein can bind to another protein, a receptor in our bodies, and there's a lot of it in our lungs, which is why we tend to get sick in our lungs. So this receptor can bind to the spike protein, and the spike protein can then bring the whole virus into the cell and to cause an infection. So that's how the virus is causing infection. But similarly, when our immune system responds to this virus, you can see the spike proteins are right here on the surface. And so of all the different proteins that are present in the virus, it would be most easy for our immune system to detect these spike proteins because they're on the outside. So for that reason, that's one of the reasons why the messenger RNA that's being delivered through the vaccine, it encodes for this spike protein. So I'm going to just show you, for if we have any immunologists in the crowd, this is a somewhat simplified version of how our immune system is responding to these particles. For the rest of you, it will probably already seem overly complicated, but the immune system is absolutely amazing in how it works. So if people like me, the drug delivery scientists, have done their jobs in creating a good particle that's able to package up our RNA and to deliver it into cells. Here's it outside of one of the cells that's going to be vaccinated in our muscle tissue. And this vaccinated cell, again, it can be a muscle cell, it can be an immune cell, or other cells that are present in the area. So this particle needs to enter our cell that's going to be vaccinated. And again, it's in that endosome. So the particle needs to escape the endosome and deliver the messenger RNA that encodes for the spike protein into this watery part of the cell. And there it can be turned into protein by the cell. So here we have the spike protein that's being made and smaller pieces. So sometimes the spike protein can get broken down into smaller pieces. All these things are called antigens. So an antigen is this foreign bit of protein that our immune system will respond to. So some of the spike protein, the cell knows it's foreign, and so it holds it out. It holds it outside its cell, and it says, hey, immune system, please come and do something about this. So there are a couple of different ways that our immune system can respond. One way is that our B cells, there will be just a couple B cells in our body that have antibodies that are specific for this particular spike protein. It's amazing how many different types of B cells we have in our body. So when a B cell finds this infected cell, or sorry, this vaccinated cell, and binds to it, it is able to produce antibodies against that particular antigen, okay? So in the event that an infection ever tries to take hold, so in the event that this virus, the SARS-CoV-2 virus, enters our bodies, our immune system will be prepared. These antibodies will have been made, and they can quickly surround this virus, bind to the different spike proteins, and when it completely coats the whole virus in antibodies, that marks this virus for clearance by the immune system. So we say it's been neutralized. All right, so that's one way this works. The vaccine teaches our B cells to make antibodies, which can then try to get rid of the virus before it ever even infects any of our cells. The other thing that can happen is either the B cell can engage in a signaling process, or there are cells called antigen presenting cells. These are immune cells that can help kick off this other part of the process. So a B cell can talk to a T cell called a helper T cell, which helps get this signaling process going. It will then talk to an antigen presenting cell. Differently or separately, the antigen presenting cell can detect some of this foreign protein on the surface of this vaccinated cell and gobble it up. One way or another, the antigen presenting cell can talk to a different type of T cell called a cytotoxic T cell. Okay, so this T cell can kill other cells. And the cytotoxic T cell is equipped now with this very tiny piece of antigen that it can use to try to match up with any cell that's been infected. So in the event some virus does get into our system, and in the event that a cell becomes infected, that infected cell will actually show pieces of the antigen on its surface, and it will match up with our cytotoxic T cell such that this cell is then killed. Okay, so there are a couple of different ways that the vaccine works, but the main takeaway here is that our B cells will make antibodies to try to sequester the virus before it infects our cells. But if that infection happens, we have this other part of the immune system over here that will make sure those cells are killed. Okay, so that's what I have, and I'm going to turn it over to Professor Dong.
Okay, Yizhou, you should be presenter now. Yeah, great.
Yeah, could you see my slides?
Yes.
Let me share the screen.
Yes.
Okay. Okay, is that good? Wonderful. So hello everyone. Also good morning, good afternoon, good evening, depending on where you are. First, I would like to thank Christine, Cara, and Fabio for their great efforts on hosting this very exciting webinar. It's really my great pleasure to share with you some examples of lipid nanoparticles for mRNA vaccines and mRNA therapeutics based on our research experience. Thanks, Katie, for the wonderful presentation about mRNA vaccines against SARS-CoV-2. I don't need to give additional background information on that. In our study, we learned that both mRNA sequences and the delivery materials are critical for an effective mRNA vaccine. On the left, here's a process for the optimization of mRNA sequences. We know the untranslated regions, UTR of mRNA, play important roles in regulating mRNA functions. So we investigated the 5' and the 3' UTR of mRNA through two approaches. One is bioinformatics analysis of endogenous gene expression. We analyzed over 4,000 different genes reported in the literature. And the other approach is de novo design, which is based on several predetermined criteria. In early 2020, we identified a pair of lead UTR after over 4 years of studies. Then this COVID-19 outbreak occurred. We decided to apply our lead mRNA to code SARS-CoV-2 antigens. Then we use our lipid nanoparticles to deliver this mRNA and induce specific antibodies against SARS-CoV-2. This slide shows one set of our results. We prepared mRNA encoding spike protein, as Katie mentioned. We then formulated this mRNA with two types of lipid nanoparticles. One is MC3, FDA-approved lipid formulation. The other one is a TT3 we developed in our lab. On the left, the y-axis, that's the specific serum IgG, and the x-axis, that's MC3 and TT3. You can see TT3 induced a high amount of specific IgG production, which is over 300-fold higher than MC3 at the same dose. On the right, we studied two different administration routes, intramuscular injection and subcutaneous injection. We can see intramuscular injection is better than subcutaneous injection in this case. For the following several slides, I borrowed from the FDA Advisory Committee meetings with Pfizer BioNTech and Moderna. These slides are about the maximum actions for mRNA-based vaccines. As Katie just described, this process involves multiple types of cells and multiple cellular pathways. I think additional studies are still needed to fully uncover the process for these COVID-19 vaccines. This slide is from the presentation of Moderna, which contains some important information a lot of researchers and the public are interested in. Does the vaccine change the host genome? They presented that only a few copies of mRNA molecule are delivered into cells, compared with over 200,000 mRNA molecules already in cells. mRNA is cleared through the natural degradation process, and importantly, there is a minimum or no chance these mRNA molecules can be reversely transcribed into DNA based on our current knowledge. In other words, these mRNA vaccines cannot alter human DNA. Here's some highlights about the key discoveries and advances about mRNA drugs. In 1961, mRNA molecules was discovered. It took over 30 years to understand the fundamental biology. Then in the year 1989, 1980, researchers started to deliver mRNA into cell and animal models. Then there's another almost 20 years of preclinical and translational studies of mRNA. In 2009, researchers reported clinical results of the first mRNA-based cancer vaccines. After that, there's over 10 years of clinical trials for a series of mRNA pipelines. Then in December 2020, two types of lipid nanoparticles formulated mRNA BNT162 and mRNA-1273 are approved by the FDA as the COVID-19 vaccines. In addition to viral vaccines, mRNA has been explored for a wide variety of applications, such as cancer immunotherapies, protein replacement therapies, genome engineering, and also genome editing. I will share with you a few examples in the following talk. So this slide shows our strategy to optimize lipid nanoparticles for mRNA delivery. We first synthesize new types of lipids or lipid derivatives, and then we formulate with several other components. Katie just mentioned helper lipid, cholesterol packed lipid, and here the therapeutic cargo is messenger RNA. Through this microfluidic-based device, they self-assemble into very nice nanoparticles, and then we can characterize the particle properties, such as particle size, particle charge, entrainment efficiency of mRNA, and also mRNA translation efficiency. We utilize the orthogonal experiment design, which enable us to efficiently identify the optimal formulation with significantly reduced experiment load. After that, we identify the optimal formulation, then we can test in different models. And here's a model disease, hemophilia. Hemophilia is a genetic disorder caused by missing or mutation of factor 8 or factor 9. Patients suffer from life-threatening bleeding or serious complications such as joint bleeds. In this study, we formulated TT3 encapsulating mRNA encoding human factor 9, and then we injected intravenously in the factor 9 knockout mice, which is a hemophilia B mouse model. So here the y-axis, that's a human factor 9 activity in MIU per ml, access different treatment groups. You can see it showed a dose-dependent recovery of the factor IX activity. At the dose of 1.1 milligram per kilogram, the factor IX activity is around 800 MIU per ml. In normal serum, the factor IX activity is between 500 to 1500 MIU per ml. That means that TT3 is able to fully recover the factor IX activity into the normal range. Recently, we collaborate with the groups of Ron Weiss and Daryl Irvine. We formulated TT3 with self-replicating RNA encoding IL-12 for cancer immunotherapy. And here's an illustration of the process. This lipid nanoparticle replicon formulation first induce immunogenic cancer cell death, and then subsequently trigger the anti-tumor immunity. Here are some results our collaborators produced. They performed a single injection of the lipid nanoparticle formulation in multiple mouse tumor models. You can see it showed a significant extension of mouse survival in B16F10 tumor models, CD26 tumor model, and also humor 1.7 tumor model. They also performed a number of different type of maximum action studies. And here is one example here. In the mid-88 sting knockout mice, the therapeutic efficacy of this formulation is significantly compromised. We are also thinking to apply lipid mRNA nanoparticles for treating other deadly disease, such as sepsis. Sepsis is a life-threatening condition caused by severe infections. It affects over 30 million people worldwide and remains the #1 cause of death. Although there's a significant progress in the intensive care and antibiotic therapy, the mortality rate is still pretty high, around 25 to 30%. And this is an illustration of our strategy. We designed a series of vitamin-derived lipid nanoparticles to deliver mRNA encoding antimicrobial components into macrophages. And then we treated septic mice with the engineered macrophages in order to boost the innate immunity, prevent bacterial immune evasion, and eliminate multi-drug resistant bacteria. In this study, we established a sepsis model by infecting mice with mixed bacteria. Here is a multi-drug resistant S. aureus and E. coli to mimic the sepsis patients. Then we treated these mice with the LNP engineered macrophages. On the left, you can see the engineered macrophages significantly reduce the bacterial load in the mice. And importantly, these engineered macrophages dramatically extended the mouse survival over 80%. And these survived mice continue to grow, and on the left, that's a body weight. And on the right, after 30 days, we noticed both white blood cells and the lymphocytes return back to the normal level. So in conclusion, I briefly shared with you the broad applications of lipid mRNA nanoparticles, such as the COVID-19 vaccines, treating genetic disorders, cancer immunotherapy, or counteracting the drug-resistant bacteria. Lastly, I would like to acknowledge my lab members. This is a group of talented postdocs, graduate students, undergrad students, and a few high school students. Thanks our collaborators for their significant contributions on several different projects. Thanks the funding agency supporting our projects. Thank you very much for your attention. I would like to take any questions. Thank you.
Thank you very much, Yizhou, and thank you, Katie. That was really great. It was really, really informative and a great seminars to really understand what's behind the technology and how we got there. So we have a few questions that we collected from the registration forms, and we also have several questions that came in during the seminars. So the first bunch of questions, we will try to group them as much as we can on a broad theme. So the first bunch of questions actually refers to the formulation formulation of the mRNA vaccines and the stability of the formulation. So you already, Katie, you already talked about the differences between the Moderna and the Pfizer-BioNTech vaccine and the differences in the lipid. We got a few questions on that, and I think you mainly answered to that, but we also got some questions about the storage temperature differences and whether this is related to the formulation and how this is related to the formulation. Oops, sorry, you are muted, sorry, there you go. Ah. Katie, sorry, you were muted, so you have to restart. Sorry about that.
Okay, you can hear me now?
Yeah.
Yes.
Okay. So the temperature is a very common question. So the temperature required for storage. So first of all, the reason why there are storage problems for lipid nanoparticles is because they are fairly unstable particles compared to a lot of other types of drugs that we use. It's because they have all those different components, and the components have come together in a very particular way at the molecular level. And so it doesn't take a whole lot in order for them to fall apart. The higher the temperature, the more the molecules move around, the more you have motion, at the molecular level. And so the easier it is for the particles to come apart. So that's why the very cold temperatures are used for some of these particles. Moderna has been in the space of vaccination for longer. I think they've had more of an opportunity to finagle with their lipid nanoparticle formulation and to add some different things. So there are different people who have found that adding sugars and some other molecules can help stabilize these particles. So Moderna has had more time, I think, to figure out how to make their lipids stable at slightly warmer temperatures in the fridge, as opposed to the Pfizer-BioNTech vaccines, which were, I think the lipid nanoparticle formulations there were developed more quickly. They didn't have as much time for all the stability testing and optimization there. And so to be safe, just to be totally safe, those are being kept at minus 70. So I think with time, so I think CureVac actually has, they're developing an mRNA vaccine that can be kept maybe in the fridge for a couple of months. Yizhou, do you know about CureVacs?
I saw some news, but I don't think it's published. I don't know. So what's the exact?
No. Yeah, it's not published yet, but it's an ongoing area of work.
Yeah.
But that's why cold temperatures are required.
All right. Thank you very much. That's good to know that there are room for improvement. And one other question that came in is so about the about the pKa of the ionizable lipid. So in the Pfizer vaccine, the pKa is 6.09. Does this surprise you, like having such a low pKa, or is this something predictable? Or
Yizhou, did you want to take this one, or I can comment?
You can go ahead.
Sure. So I can't say it really surprises me. It's within a range that I think could work. So what we care more about, I think, in terms of pKa is the pKa of the surface of the nanoparticle itself. So that's really what is being shown to the endosome and to the endosomal membrane inside the endosome, not individual lipids that are, ionizing themselves. The whole particle is changing as a function of pH. And so, without doing experiments on their exact lipid nanoparticle, my expectation is that when that particular lipid is in the context of the LNP, that it is taking on sufficient charge while the pH is dropping inside the endosome to facilitate that escape process.
Right.
Totally agree. I think the pKa is very important the beginning for the formulation of the nanoparticles, because under acidic condition, they need to interact with negative charged mRNA molecules. After the formulation is constructed, then they need to interact with the cell surface and then go through the endosome pathway and endosome escape to release the mRNA molecule to the cytosol. Then it can have the function. So that process is very important because the endosome pH is changing, like from the early endosome to late endosome and lysosome. So that makes the pK is a very important parameter to tune for the endosome escape.
Fantastic, thank you. And someone from the audience right now asked, what's the role and what if the OH group, head group in the lipids formulation is necessary for mRNA delivery? What is the role of it?
It's not necessary. So there are plenty of examples of lipids, ionizable lipids that are used that don't have that alcohol group, but it's also acceptable. So it happens to appear in these formulations.
Yeah, I think it's more about the structure as a whole. So like for MC3, it's FDA approved formulation. It doesn't have hydroxy group on the molecular. And for this molecular hydroxy group, I think it facilitates hydrogen bonding, interact with other formulation components. But like Katie mentioned, it's not essential component. It has to be there. It's more like the overall property of the nanoparticle.
Great, thank you. There is, I think, another couple of questions about the formulation, and then I'll hand over to Christine for another bunch of questions. So another one is like, does it matter how much mRNA is contained in the lipid nanoparticle, and does the size of the particle have an effect on the dosage given?
I don't think
take that one? Yeah.
Exact techniques to quantify how many more mRNA molecular copies in each nanoparticle, but based on the estimation, from the industry and also academic labs, we think there's only very small number of copies in each nanoparticle. And also, I think that varies between particle to particle because the particle is not like a single size. It's a range. I think for the presentation from the companies, they presented the particle size for the current COVID-19 mRNA vaccines is around 80 to 100 nanometers. So it can be varied for each nanoparticle. And for the size, I think it's important, but I don't think there's a direct correlation with the delivery.
And one thing to note is that, ultimately, it doesn't matter. I don't think it matters a lot whether we have fewer mRNA in slightly smaller particles or a little bit more mRNA in slightly bigger particles, because ultimately, when we talk about dosing of the vaccine, it's based on the amount of mRNA in the formulation. So a specific amount is going to be injected into your arm, and so might be in slightly bigger or slightly smaller nanoparticles, but ultimately that shouldn't affect the efficacy too much.
Yeah, I think one parameter is encapsulation efficiency. So for the total mRNA dose, I think the companies are also academic labs always characterize the encapsulation efficiency of mRNA, let's say 90% or 95%. So that's an important factor to determine. So if there's a lot of free mRNA, then they have much less chance to have the desired function. If a majority already encapsulated in the nanoparticles, then they have better chance to be delivered and have the function.
I suspect that the encapsulation for both of these lipids is very high.
Yeah.
Thank you. There is one other question about the oxidation. How vulnerable are the lipids? How much vulnerable are these lipids toward oxidation? And if they are oxidized, what would be the fate of the mRNA loaded into the nanoparticles?
So
I'll let the chemist take this one.
For the molecular structure itself, I don't think it's easy to be oxidized. And for the ionizable lipid, Katie showed Pfizer, Moderna, and I think both are relatively stable. I think for storage, I think the main storage condition is trying to optimize it for the particle storage.
Okay, I think I can hand it over to you, Kristen, for the other bunch of questions.
Yeah, thank you. So lots of people were interested into the delivery route, so intramuscular injection versus other delivery routes, and which kind of cells can actually take up these nanoparticles, and how is this affected by the delivery route? That's a few people have asked this.
So I covered that a bit in my presentation, but the short answer is that these particles don't have any inherent ability to exclusively go into one cell type versus another. So they're injected right into the muscle tissue. The cells that are present there are essentially sitting ducks. So if they are inclined to take up these particles, they are given the opportunity to do so because they are basically bathed in these nanoparticles. So the muscle cells are, they're transfected, which is what we call when the material gets into the cell. They're transfected to some extent. The immune cells that are present in the muscle tissue, there are a lot of them. That's why, that's in part why all vaccines are given in our arms, because there are lots of immune cells there that can respond. So those immune cells will also be able to take up some of the vaccine. And, to my knowledge, there aren't any studies that have completely decoupled the effect yet of delivery of the vaccine to, say, the muscle cells versus the immune cells. But I think it's fair to expect that both of them play a role. The immune cells do kind of what I showed, where they're like actively responding and talking to the other immune cells, whereas the muscle cells, they'll still make the antigen. They'll still show it to the immune system. They can't do as many things as the immune cells do when they uptake the particles, but they can still help.
Thank you. And following up on this, we had a question because you showed how the T cell side of the immune response, how the killer T cells can then recognize or can present the antigen. And somebody asked if the killer T cells would then also kill the vaccinated muscle cells because they present the antigen.
Yizhou, do you know the answer to that?
I don't think so, because the body has its own recognition process. Generally, muscle cells, if they present antigen, then there will be the T cells with them. So, and for cytotoxic T cells, I think they mainly try to clear infected cells. So I don't think there's a worry on that part.
My very basic understanding is that the way an infected cell would show the antigen is going to be different than the way a vaccinated cell would. And so, that might have to do with the differences. We would really need an immunologist to step in here.
People were also very interested about the clearance mechanism. So what happens after it's taken up and released the mRNA? What happens to the lipids and to the lipid nanoparticles afterwards?
So the lipids, as I mentioned, they'll degrade to some extent. And so they'll be small enough that they can be flushed out of the body. The body wouldn't really have additional use for those sorts of materials. The kidney is able to clear molecules that are that small pretty effectively. And as for the other materials, so phospholipids, cholesterol, they're all naturally occurring materials in our body. And so, you know, I don't know, maybe the body could even like reuse them for certain things, or you know, we're not super worried about that. The mRNA will be degraded. It gets degraded eventually inside the cell. It's a temporary molecule. It only hangs around for so long.
And some people were also wondering what you guys think, what is the most significant bottleneck of the technology of lipid-based mRNA delivery at the moment?
I think now the COVID mRNA vaccine, it's really a milestone and a proof of concept. I think that's a really huge breakthrough, and also that can help like well control this current pandemic. So that really opens the opportunity for many other therapeutic applications. I think we have to match the delivery to therapeutic indications. I think similar to the previous question, so what cell type we try to target, we try to deliver, and what function we try to either restore or correct. So that's very important. If we can deliver the cargo or mRNA to very specific cell population or disease cell population and rescue the phenotype, I think that would be very important. And also, all these biotech companies, they have quite broad pipelines. There are many like therapeutic indications in clinical trials ongoing.
Wonderful.
I would just add, as far as the bottleneck is concerned, I think, you know, the delivery science is clearly at a place that it can support therapeutic development. I think we have a lot of questions, you know, as Yizhou mentioned here, about how exactly the delivery is happening and what cells specifically, but these particles work well enough to make this happen. And like, how amazing that scientists could work so hard and go from the sequence to first in human within a couple of months. It's amazing. So that part of all of this is here. If we had some other pandemic, or in the event that some variants really do require additional mRNA sequences to be incorporated into the particles. The real bottleneck, as I'm sure many of you are aware in the news, is on the manufacturing side. And then also the stability side, right? So the stability isn't so much of an issue for those of us in first world countries where we have refrigerators, so-called cold chain storage available to us. But this is a problem for people in the third world and places where you need to transport, where there just isn't cold storage available. So that's one of the main concerns. And then also manufacturing. We need more rapid production of these lipid nanoparticles.
Great, thanks so much. Chiara, I think I'm gonna hand back to you then for the next bunch
Yeah,
of questions.
absolutely. So yeah, so actually those were questions that were asked about the versatility of the platform, and as well about the scalability and the manufacturing issues with that. Another set of questions that came out, and I think that might be worrying actually people that read news and look at the news, is the adverse reactions that have been reported for some people. So a lot of questions were about the effect of PEG, and what's the allergic reaction, is it caused by PEG? And there are other, as Katie was saying at the beginning, there are others FDA approved formulations that contain PEG, and yet these severe allergic reactions had not been reported, or had not been, didn't make the news that much. So can you discuss this a little bit? Is there will PEG, and also will the rise of PEG antibodies somehow threat the widespread use of lipid nanoparticle deliveries for other drugs as well?
So first, I just want to make sure everybody is aware that the you know, the news has really focused on these vaccines. So the number of adverse effects and the types of adverse effects being seen are, you know, within the same kind of level as what we would typically see with a vaccine. There are always adverse effects. It's just that when people go in and they get their chickenpox vaccine or their annual flu vaccine, the media doesn't think it's very interesting if somebody has an allergic reaction. And so there aren't news stories all over the place. You know, woman in Alaska has a severe allergic reaction to the flu vaccine. You know, we just don't see that. So all of this, you know, we see it, and so we think it's more of a problem than it normally is. So just to put that in perspective. That being said, yes, it's always worrisome when there are severe allergic reactions. There are so many people, there's so much variation in the way that our immune systems respond to different things in our environment. It's why some people have allergies. It's why some people have autoimmune diseases. It's because some of our immune systems, they don't work quite right. So it's really hard to predict when these specific particles are the thing that's going to upset somebody's immune system, because there are just so many different people. The PEG. The PEG could be causing the problem. Some anti-PEG antibodies have been found after administration of other therapeutics. That being said, you know, so my lab has been looking at this for a while. We haven't published any of it yet, but what we're seeing is that the creation of antibodies in response to PEG in lipid nanoparticles, it actually has a lot to do with the totality of the lipid chemistry and the totality of how the lipid nanoparticle is being seen by the rest of the body. So for those of you who know what toll-like receptors are, there's a toll-like receptor 4 that, you know, can essentially recognize some of these lipids as foreign. And if that happens, it can set off a process whereby our immune system responds to the PEG. So it's all this very complicated dance between the innate immune response and our adaptive immune response. You're not always going to have antibodies to PEG, but even if you don't have a response in 99.8% of the population, it's still possible. It'll happen for others.
Yeah, I agree. I think that need to be carefully studied. PEG, I think it's one component that need to be carefully studied, and I saw the news, the companies are investigating on that. And also, I think it has to be studied under the right context. I think in previous clinical reports, there are many other medicines with a PEG component. The researchers reported it's always case by case, depending on the molecular weight of the PEG, dose of the PEG, and particular administrations. So that's, I think, need to be carefully studied with all these parameters for the current COVID-19 vaccines.
I also just want to add one thing that I think is important to keep in mind. So a lot of times delivery systems and these nanoparticles aren't actually created with the best materials in terms of delivery or efficacy. Sometimes the choices are made for other reasons. So we're not all doomed if PEG eventually causes antibodies in a lot of people. There are many, many alternative chemistries that can be used in place of PEG, some that are actually better, even better than PEG, at preventing immune cell uptake and some of the other things that we worry about with normal PEG-related medicines. It's just that PEG is FDA approved. So it's an approved molecule. And so whenever you're developing a new medicine, it has to go through the FDA, and the FDA is much more comfortable with anything that's already been approved as opposed to anything that isn't. And you know, that's why a number of the different ingredients in these lipid nanoparticles were chosen. Okay, so the helper lipid that's in there, it's not usually the best for messenger RNA delivery. It's just that it was already FDA approved in the first lipid nanoparticle formulation from Alnylam for short interfering RNA therapy. So it's already been through the ringer, and that's why it's being used again. So we do still have hope, is all I'm trying to say, if PEG does ultimately pose some problem.
That's great, because actually one of the other questions that came up was like, are there alternatives out there that have been studying to prevent this effect, or in case this effect becomes a real problem? Thank you so much. Yeah, and then I think Christine has a few questions on delivery, actually, alternative delivery strategies.
Yes. Lots of question came in about targeting of these lipid nanoparticles, because the nanoparticles that are now used in these mRNA vaccines, they're not targeted towards a specific cell type. And people were wondering whether there are options to target these lipid nanoparticles, if they could be improved, if they could be modified somehow to target specific cells.
Yeah, I think definitely. So in the literature or previous studies, there are many strategies developed targeted delivery, not just for mRNA-based nanoparticles, or like for many other different type particles. Researchers installed like targeting ligands, antibodies, small moleculars, aptamers, all these on different type of targeting different type of the cell populations. And I think, like I mentioned before, it has to be matched to the specific therapeutic indications. So for this particular case, for COVID-19 vaccine, I think maybe it's better to have broad cell population to express the antigen, and then antigen presenting cells will have better chance to interact with these antigens. So for other applications, probably it will be better to target specific cell populations and have better efficacy and reduce the toxicity.
And I'll just add, and I agree, I don't see any point in targeting these mRNA formulations unless at some point we figure out that, hey, it's like way better for the immune cells to be transfected and we don't need very much in the muscle, or vice versa. I don't see any reason to actually target these right now. For other applications that are, injected IV, where you have things circulating, traditional targeting mechanisms where you put ligands or something like this, other proteins on the surface of your particles, they've had very limited success. It's often because the particles still aren't like passing by the particular tissue type that needs to be targeted. And so one cool thing about these lipid nanoparticles is that they sometimes have what I refer to as endogenous targeting capacity. So when you inject them into different areas in the body, there are proteins that are present, say, in our blood or in the extracellular space in our muscle tissues, and those proteins can absorb to the surface of the nanoparticle. And there's a significant amount of research now that shows that depending on which proteins absorb to the surface, it actually influences where these particles go and what cells they enter. So for this reason, I think sometimes you don't even have to put things, decorate the surface with different ligands. You just need to change your chemistry. You need to change the surface of the nanoparticle in a way that those particles will absorb different things. It's called a protein corona, onto their surface and take them to the cells that you want. But there's still so much to be learned in that area and what types of protein coronas you need to form to get to certain types of tissues.
So people were also wondering about the advantages of lipid nanoparticles compared to other delivery platforms or strategies. I think, Yizhou, you mentioned it in your talk a little bit, there are different strategies that are explored. And so what's the advantages of lipid nanoparticles? And for example, compared also to to inactivated viral viruses as delivery vehicles?
So I think lipid nanoparticles, like Katie mentioned, it has been studied for many, many decades. So researchers at academia or industry, they have better understanding of the property. And because this is a pandemic, researchers can assemble the particles and the mRNA very fast. And then we need to move to the clinical trial within a relatively short period of time. But meanwhile, there are many particles ongoing in the preclinical studies or even in clinical studies, like polymers, protein-based nanoparticles, even like some inorganic particles, yeah, so all these, I think, at a different stage of the studies, they all like show very promising results in the literature. So it just, I think, lipid nanoparticle has been studied for quite a long time. It's well established for the properties and for the production.
And I have to just add, I have to laugh when I see in these news reports, people, kind of freaking out about like how new the technology is, like this crazy new technology. We're using lipid nanoparticles because researchers have been studying them for decades. And so if you asked somebody in delivery science if lipid nanoparticles are new, they would say that is a snoozer. I mean, I have publications rejected because people are like, lipid nanoparticles, boring, so boring. So the polymers and these protein particles and like protamine and other things, like these are all the newcomers. Now, those are new, right? More new anyway.
I just want to add one point. I think there's a broad chemical space we can study, either lipids or polymers, and the new chemistries are developed. They can be applied for many different indications. People refer to lipid nanoparticles or some other polymer particles. It's very general, very broad. In fact, the properties for current nanoparticles is significantly different from the early days because we are delivering very different therapeutic cargoes and using very different chemistry and targeting strategies.
Yeah. I guess another question that's been touched upon a little bit, but I think it's quite interesting. Is it how difficult is it to improve the stability of the formulation? For example, to not so that it doesn't have to be stored at the temperature, or that it could maybe circumvent the cold chain to a certain extent. Is this a very difficult thing to do, or is this something that could be fairly easily done? It just hasn't been done yet because we're in the middle of a pandemic, or how would that work?
I think probably it's like between the science and art for this type of like optimization, because there are many parameters we can tune to stabilize the particles, and the pharmaceutical industry has been using all kinds of excipients to stabilize different type of therapeutic agents. I think sucrose currently used in the formulations already used for many years, and there are many other candidates I think can be applied to stabilize this. You just need a little bit more time to find the optimal condition.
I'll give the short answer. I think it's difficult.
All right. It is indeed. Okay. Chiara, maybe in the interest of time, we move on to the next parts that we get through all of them.
Absolutely. So a lot of questions came in about how easy it would be to modify the vaccines to actually face other possible pandemics or other possible. Let's stick to this one. Other possible mutations on the COVID, on the SARS-CoV-2, and is it possible also to eventually combine different mRNAs that could kind of like cover the whole family as much as possible?
Yeah, I think that's the beauty of this mRNA technology, right? So within a few weeks, figure out the sequence, then they produce the mRNA and formulate particles, and within like a couple months, they can initiate the clinical trial. So I saw the news, the companies already making vaccines specific for the mutated viruses. So this is a big advantage that can be tuned very fast for different type of emerging pathogens.
It's so exciting. I mean, it's one of the most exciting things about messenger RNA therapeutics in general, and particularly their use in vaccines. It's completely a platform technology, very modular. So you can just swap out different messenger RNAs because they're all made of the same building blocks. They look very similar to the lipid nanoparticle, and you can keep the lipid nanoparticle the same. It's just like super exciting from a translational perspective.
Yeah, I just want to add one point. It can be combined with a cocktail mRNA, then like against a different type of virus or mutated virus.
That's really cool. That's actually really interesting, and really it's when they first got approved, I think all of us kind of like breathed that, because like knowing that they could be easily modified, it's something that just gives a little bit more hope for the whole situation. So
And
you actually yeah, sorry.
Can I just mention that, like, the traditional vaccines that we're used to receiving, you know, for example, the subunit vaccines where people are producing very specific proteins and antigens, you know, unfortunately, that's one of the challenges, is that every time you need a new protein that's you know, different, you have to go through this process again. So that's not to say they can't do it quickly. They did it quickly. You know, they did it quickly this past year. It's just something that's going to be harder for that type of vaccine versus mRNA.
Right. So in your opinion, which will be next big, the next big thing on mRNA vaccines? So, I mean, we are in the middle of a really big thing, so I think that there is a time. But what are the other diseases that can be tackled with this type of technology, and what's in the pipeline, if you know, or what's been approved or close to be approved that use the same technology?
I think first it's proof of concept with the vaccine, viral vaccine, and in the pipelines there are quite a few pipelines for like controlling other type of viruses. I think probably they have a better chance to be approved sooner than before. And also in the pipelines there are quite a few candidates like we just mentioned the cancer immunotherapies. That's a big family of diseases and therapeutic agents. And also protein replacement therapy, gene editing. I think all these are ongoing. Then it's hard to predict which will be the first one.
In the vaccine space, I mean, people are studying, you know, at least at the academic level, they're studying using mRNA vaccines for the flu, for HIV, for Ebola, for Zika, for a number of different viruses. So flu to me seems like, you know, this is just speculation. I'm not, you know, I'm not on the industry side of things, but flu, you have, you know, you need to develop new flu vaccines every single year because of the evolution of the viruses, because of how modular mRNA therapeutics are. I think that would be one very nice application of the technology.
Great. Actually, talking about the flu vaccine, someone sent a question regarding the whether you think it would be possible eventually to have a vaccine that is injected nasally, so like the flu at the moment. Is this something that the researchers are working on?
Not sure anybody's working on that yet for SARS-CoV-2. Maybe somebody is who's very forward thinking. I think right now, like, we're not worried about alternative, you know, more patient-friendly ways of vaccinating. We just want to go with the tried and true. Yizhou, do you know of anybody who's looking at pulmonary delivery?
Yes, I saw some published work or like deposited into bioarchive. There are some studies. I think it's great. So because we are always having emerging pathogens for future pathogens, if we have better administration route, that would be even nicer.
That's really cool. Yeah, so we actually received a lot of other questions. I'm not sure we'll have the time to go through all of them. What do you think? Is there anything that caught your attention, Christine, specifically, or should we just?
I think we've gone through quite a lot of them or touched upon them in the different discussion points. I think I guess to maybe wrap it up a little bit, I think we had a few questions about the future of the technology that we touched upon and what this means, for example, for the treatment of other diseases. You mentioned cancer, immunotherapy, but for example, also for genetic diseases and things like that. What is the outlook? Are you guys both now super optimistic that, okay, now this is going to go much faster into the clinic and all the decade-long research is going to translate faster, or do you think this is still a huge hurdle to overcome before it can actually be applied for other diseases regularly in the clinic?
I think, like I mentioned, this is a big breakthrough based on the COVID-19 vaccines that really promotes the field and both like academia and the industry to expand the research opportunity on many different therapeutic indications. Although we have been working on quite diverse, either the delivery system or diverse therapeutic indications, but I think in the future there will be more and more like demanding for other type of diseases, and also new type of technologies will be developed based on the current success.
So I'll just comment. So the first lipid nanoparticle, the first RNA drug that was approved was from Alnylam. It was approved a couple of years ago for an orphan condition, or it was given this status, and it's being used in very few people. So very few people have the condition that's treated with this type of lipid nanoparticle. So the previous kind of human data that's out there on the use of lipid nanoparticles, it's been very sparse, which meant that any additional indication or any additional therapeutic that was going to come out was going to require a lot of scrutiny because there was no understanding of how this particle, this type of delivery system would behave in a broader population. We have now been faced with literally a disaster, an absolute emergency, where the hands of our regulatory agencies have essentially been forced into these emergency use authorizations because the lipid nanoparticles seem safer than coming down with COVID-19. But the result of this is that we are going to have a huge amount of data, millions of people who will have been given these lipid nanoparticles. We will have a much better understanding of how they behave broadly in all sorts of populations, young and old, people from different backgrounds, different ethnicities, different parts of the world. And so with all that data, I think the regulatory agencies are going to be far more comfortable. They're still going to be cautious, of course, as they always are with new medications, but they're going to be far more comfortable with the deployment of these sorts of technologies in the future, which I think is just, absolutely wonderful. Because regulatory agencies haven't known what to do with these sorts of drugs before. All of our medicines used to be kind of single molecules or not very complex molecules. And the FDA in particular, and I'm sure other regulatory agencies around the world, they've had to figure out how they're going to handle these sorts of nanoparticles that have like 5 different ingredients, all these new types of manufacturing. How do they actually assess and evaluate the safety of these sorts of medicines? So it's something that's evolving and a slow process, but I really do think that this emergency, while we certainly would have preferred not to have it, I think it's really going to open the doors for more implementation of this mRNA therapy in the future.
Great.
Let me just add one more last point. So I think we are all quite optimistic about the future of mRNA therapeutics, and there will be more and more mRNA-based drugs approved in the future. But I also think please do not be over-optimistic. So mRNA is just a therapeutic agent. It cannot solve all the disease or all the issues. I already got some questions from other people. This maybe solve all the disease and cure everything. So I think we need to be cautious on that.
Yes, I think
Mhm.
that's a wonderful wrap up. Optimism with caution. I think that sounds good. Chiara, I think we've gone through as
I think
many
we've gone through the more, I mean, the
most
most burning questions
yeah
and the most general ones. Yeah,
yes.
absolutely.
Wonderful. So thank you both so much for the talks and for taking the
Thank
time.
you.
And thanks everybody for listening. We hope that we could shed some light on the technology, and I hope people now share a bit of our optimism for this technology. As Katie mentioned, it's been with the nanotech and material science community for quite a while. So so thank you everybody for joining us today, and please everybody stay healthy, stay safe, and have a wonderful rest of the day. And thank you everybody.
Bye, everybody.